bit of a dilemma always with the mic because I want to move around, but I also wave my hands. So it's kind of one or the other, so I pick moving around. It's great to be here after Rebecca because we're just going to move a bit further in the family tree of plants. So plant cells, not seeds. That was something brilliant that I did not come up with. I think it was Isha who came up with it. Thank you. I just stole it from you. So plants, indeed, they do the heavy lifting in our food system, plants and algae. They are the primary producers who actually make the biomass that we eat and that we feed to our animal cell cultures. But they also produce a lot of other stuff. They also produce the important biomolecules, the complex chemicals that we need from our food as vitamins or antioxidants or you name it. And plants are great in making those things. I mean, they have to. They cannot run away from the sun. They cannot run away from things that try to eat them. They have to protect themselves chemically. And obviously our diet is based on plants. And there is roughly 3,000 plants that we have used or use in our diet, but there's only 8 that form the basis of our diet. There's 8 plants where we get more than half of our calories from. So that's quite a narrow part of the plant kingdom. In addition to those 8 major crops, there is something like 30 fruits and vegetables and berries that we use a lot as humanity. But then there's a lot of things that we're not using for one reason or another. This is one of them. This is Arctic bramble. It's a delicious berry. Grows in the Nordic environment of, say, Finland. It makes only one berry, one single berry per plant, and it cannot be cultivated. So it's a real sucker. I mean, it tastes good, but you can't get them. Yeah, go to the forest with the mosquitoes and pick some if you want to eat them. So could cellular agriculture do some good here? Maybe it could, but I have to talk through a bit of history to get there. So the biggest difference between plant cells and animal cells is that plant cells are totipotent. So any plant cell can de-differentiate and differentiate again to pretty much any role of the plant, and that's why you can take cuttings from plants. You can take a twig, stick it in soil, and it will grow new roots and new shoots. So this was figured out already quite a long time ago. Gottlieb Haberland was a guy who's quite often cited as the father of plant cell and tissue culture. He figured out that it would be possible, probably, to grow plant organs or cells in vitro infinitely. That was already beginning of the 20th century. He got the idea right, but he didn't quite get the technology right. It took like almost 4 more decades to grow the first cell cultures in vitro. And those were actually carrot and tobacco cells. This is a cut slice from beetroot that I did. And you can see here on the edge, there is some de-differentiated cells, we call them callus, starting to grow. So those are the plant cells that don't know what they will become when they grow up, shoots or roots or something in between. It took another more than a decade before those cells could be cultivated in suspension culture, in liquid culture. These are tobacco cells. They're also called the HeLa cells of higher plants. They are really the workhorse of plant physiology. And we can thank them for a lot of stuff that we know about plants and we know about cells and life in general. Another big difference between animal cells and plant cells is that plant cells require very, very simple media. Some minerals, sugar, that's pretty much it. No serum, none of that stuff. Sometimes we use our secret sauce is coconut water. For some very tricky things, add coconut water in there. We have no idea why. It works. It's like serum. All right, so suspension cultures in 50s, and it took only 1 year after that when the first whole plant was grown from a single cell. So I think that tells a whole story about this totipotency. You can differentiate a complete plant from a single cell. And this is what the plant cell culture technology is used a lot nowadays. For example, most of the orchids you buy are propagated from cell cultures and then differentiated into plants. That's how you can get rid of a lot of plant diseases. You can also propagate a lot of clones from very, very small starting material. Another application is production of very complicated chemicals like Paclitaxel. It's one of the most used cancer drugs in the world. Originally extracted from the bark of Pacific yew, and that's not a very sustainable source of a pharmaceutical, because you would first of all have to cut all of them to get enough, and also the weather influences a lot the amount of material and the quality of the material you can get. It took a long time, but for a couple of decades now, Paclitaxel has been produced in cell cultures in very big bioreactors. So fast forward quite a long time. That's me starting to work as a PhD student at VTT. VTT is one of the biggest research institutes in Northern Europe. We do anything from fusion reactors to autonomous vehicles to food tech to production of pharmaceuticals, everything. And especially we have a long history in doing research on plant cell cultures. On the other hand, we have history in producing industrial enzymes for paper and pulp industry. So my job as a PhD student was to figure out how to use these tobacco cells to produce complex recombinant proteins. So here's my favorite. This is a tobacco cell line producing something that has also a green fluorescent component in it. It produces the protein more than any other plant cell line ever in history, a gram per liter. I had to rename the cell line as Hulk because it's quite incredible. It's very, very green. So another part of my PhD was to figure out how to grow these tobacco cells in industrially relevant scale. That's another thing why VTT is an awesome place to work, because we have this candy store where we have more than 60 different bioreactors up to a scale of 1,000 liters, where we grow those plant cells. So I could figure out pretty neat ways how to do that in scale. And what's surprising is that plant cells are quite happy with many kinds of bioreactors. They grow pretty happily in pretty much whatever bucket you put them in. All right, back to these things. Industrial enzymes, complex molecules in plant cells, but what our group has been mostly interested in are those valuable biomolecules from the Arctic nature, from these berries, and how we could use those in some sort of reasonable way. So we have this huge culture collection. We have these different kind of plant cell cultures grown in the lab. And I was working with tobacco cells. I mean, these are interesting. It's always greener on the other side, or more red, or whatever color. And these have been used, obviously most of the people in our team come from pharmacological background. So they were interested about the health benefits. And the biggest industries that we have been working with for a long time have been cosmetic industry. For example, Lumene is one of the Finnish cosmetic brands, and we produce the Cloudberry cell cultures for their products. So Cloudberry cell cultures, they produce pretty much the same biomolecules as the cloudberries, which are hard to get. So we grow them in tanks and provide it to these cosmetic companies. But, I mean, take another look. These things look yummy. So pretty much the first question I had when I came to the lab was, where's the coffee? And next was, what do those things taste like? Can you eat them? I mean, if you're growing strawberry cells in the lab, what do they taste like? And the guy said that, no, you're not supposed to eat anything in the lab. They come from the pharmaceutical industry. That's completely understandable. I did not. So it was a long struggle. That after all, we started to play around with the idea of Berry 2.0. What if you could grow plant cells as fresh food? I mean, they do produce many of those interesting compounds that are important for our health in many ways. So why not just eating them? One of the problems, obviously, was that you need to get the price down quite a lot to be able to produce them in any reasonable way. But good news is that they are happy with all those buckets. This is a wave bioreactor designed originally for animal cells. Plant cells are happy in it, but that's way too complicated for them. So we started to think about simpler and simpler ways of propagating these cells to get the, production easier. And at this point we did what any reasonable, self-respecting research group should always do: hire a designer. This is Nico. Nico is right there. Came into our team to do his master thesis in design with bioreactors. So we started to think about, how could you redefine the whole concept of a bioreactor, the whole thing, what it means to grow cells. And Nico did some brilliant work in designing the whole concept around it. What would it look like if you could actually grow these at home yourself? And so came out the home cultivator, home bioreactor. It was called CellPot before we figured out that there was a company making flower pots with the same trademark, so we had to give up that name. Marianne talked yesterday about the idea of Nespresso machine. So we kind of went to that approach. So you take a capsule with cells and some nutrients in there, pop it in a device that looks like a design lamp, fill the tank with water, turn it on, wait for a week, and then you harvest your ½ kilo of fresh Arctic bramble cells, or strawberry cells, or lingonberry cells, or crowberry cells, or name it. A fascinating thought. You could do it at home, but it doesn't need to be at home. It could be something like a microbrewery hipster kind of thing somewhere where you go and you get your berries. It could be a big industrial bioreactor where you get your stuff. But I think this is quite a fascinating approach that you could do it at home. So these pictures are really, really screwed up. Well, there's a couple of more of those spoons. The most important spoon is here on the right. So at this point, many people are asking, so what do they actually taste like? For the first year, I had to say that pretty much like nothing. They have this bit sandy texture and they taste like nothing. And obviously, the food people were just thrilled. They don't taste bad. That's very, very good news. I was not that thrilled. I wanted them to taste really good. It took us some time, but quite recently we figured out how to kind of process them a little bit, make kind of jam out of the cells. So we came up with something from lingonberries that tastes like lingonberry curd or lingonberry jam. It's quite delicious. The problem is that when you make a jam out of strawberry cells, it tastes exactly the same. Good news is that when you make jam out of green strawberry cells, it tastes like lettuce. So it kind of doesn't add up. Luckily, we have something like 30 more plant cell lines to go through. I'm expecting that there's something interesting coming out there. But we're really in the beginning of designing or engineering the flavor of the cell cultures themselves, because that was not in the scope for those 20 years that we have been working with those, and also how to process them into something that tastes good. So where we're right now is that we have funding. So we are a research institute. We're not a company. We are a research company, but we're not a plant cell company. We have funding from the Finnish Innovation Fund to do work on the concept and figure out what it is actually, what is actually the product, what is actually the concept that we're building, and what kind of a problem does this really solve in the world, and who are the partners that we want to build this with. So if you have any great ideas, come and talk to me. We should figure this out by the end of 2018. If we can figure this out today, even better. So I want to kind of leave you with one thought, and that's here. So what if instead of having extinction of species, we would have like extinct gin made of those species? Wouldn't that be exciting? Thank you.
All right. So we have questions populating the slider. Thank you for your talk. It was amazing. Do you see an application of plant cell cultures with CO2 recycling?
No.
No. And why not?
Because these plant cell cultures are not photosynthetic. We need to give them sugar. So in that sense, no. But I see applications with some other organisms. I mean, algae, obviously. I think we're going to hear like next something more about recycling CO2 with some microorganisms. But plant cells, no. They need sugar. We need to feed them with something. They don't capture the CO2.
Excellent. In what ways do plant cell cultures provide that animal cultures do not? That's a very broad question, I guess, open to interpretation.
So plant cells cannot provide the protein. To be quite honest, I don't believe they can provide the bulk food for humanity. They will not solve the hunger problem. But they can provide many of those complex biomolecules that we need for our food. They can provide many of those phenolic compounds that we need for our health. Those you won't get from your animal cell cultures. Or if you do, then you need to feed that stuff for those cell cultures. Yeah. And that's tricky. But plant cells are really good at making very complex molecules from very, very simple molecules. So in a way, they are cell factories creating complexity out of simplicity.
Excellent. Could the easier nature of culturing plants over culturing animal products make it compatible with new urban farming trends? And I guess that also presupposes that you consider culturing plants easier.
Yeah, absolutely. I mean, I think, that's something that we're using. I mean, growing animal cells is a very, very complicated thing. Mm-hmm. You know it better than many people in this world. Growing plant cells is very easy. So that's why we can even play with the idea of taking that into the hands of the consumer, mm-hmm. taking it to your home, to a kitchen counter, because it's so easy. And I think that could be kind of in the broader sense, that could be kind of a gateway thing to accept cellular agriculture in more general. Yeah. So I think in that sense it could serve also a bigger purpose here.
I like this one. Could plant cells expressing growth factors be co-cultured with animal cells to promote growth and differentiation?
Yes. Why not? Yes, we can express animal proteins in cell cultures. Absolutely. Yeah, I never really thought about that. Yeah. Absolutely. In general, I would like to see this more complex system of various co-cultures feeding one each other, building this ecosystem of bioreactors. That would be academically very interesting.
Yeah. And, well, that kind of goes back to what Kate was explaining yesterday about culture media, and that manufacturing all the parts might not be the best answer, but co-culturing could be. And so just to follow on with that, I'm curious scientifically, what are some of the benefits that animal culture could see from co-culturing with plant cells? And what might be a plant cell that you would start with to start exploring that concept?
I would kind of turn it around a bit. Mm-hmm. So there is several components that you need in the animal cell culture that are growth factors or hormones or other animal products of animal origin. What if we could grow plants that could produce those compounds? So it doesn't necessarily need to be a cell culture. It could be a plant grown in a greenhouse, plant grown in the field, producing those animal proteins. One of the interesting compounds used in the cell cultures is transferrin. So the transferring protein, transferring iron, the stuff that you can buy from Sigma, if I got it right, is produced in transgenic rice grown on the field. So you can use these plants grown in the field, produce those animal proteins that you might need in your serum-free culture media.
Very nice. I like this latest question. Maybe I missed it, but what's the advantage of using the cell over the seed?
So yeah, surely. In many ways it's easier to plant the seed and grow the plant. That's very easy. But in some cases you just cannot. If you try to grow the Arctic bramble, you just cannot farm them. They are very susceptible for many diseases, and they just are not suitable for farming. So there's a lot of plant species that we have not domesticated, and we probably cannot domesticate. But planting the cells allows us to take advantage of those very complex chemical or biological machineries that those organisms have and use them without planting actually the seeds.
Excellent. Can you use plant cells as a nutrient source to increase efficiency and yield for conventional farming? So it says either in soil or hydroponics.
I'm not sure if I'm getting it. So use plants as nutrient source to increase efficiency and yield in conventional farming.
So using plant cells as a food source for other plants, maybe?
I wouldn't. Like compost. It's a very expensive way to make compost. We have, yes. I would say that at this moment, no. The benefit of plant cell cultures for the conventional agriculture, conventional plant production is in the plant breeding aspect of it. And they're very useful as a tool in the process to breed those plants that we can use in those conventional systems. But in the production, hard to see the advantage that there would be, hard to see a way. I don't know, if somebody has an idea, just come and tell me. Let's figure out and make it happen.
I kind of like this one. What do you think about the public's embrace of superfoods? And could you use that as a strategy to get people to eat new formulations of plant cells, so like cell jam?
Yes, absolutely. Now, this is obviously controversial as heck. I mean, I don't really like the concept of superfoods, but there is many who do like it. Yeah, I can see that it would be easy to sell cell jam as a superfood. But I would be quite conscious with if it actually is super. Is it better than? Some might use it and consume it because it's something new, something special. But I really want to have something real there. It needs to have some flavor, something that is not there in other foods. Or then it needs to be really significantly healthier, better than something else. Superfood is used really easily. In that sense, I don't really... Yeah, could be a marketing trick, but I wouldn't use it easily.
It almost feels like cheating.
It feels a bit like cheating, yeah.
A bit like cheating,
it needs to be really super, otherwise I wouldn't use it, no.
That makes sense. So where do you think plant flavor comes from? Like, why does a green strawberry taste like lettuce? Does it have the same compounds in it that lettuce has?
It could be. That's a really good question. So many of these plant cell cultures, they produce the same complex molecules, so the same flavor compounds that the berries themselves produce, but they do kind of have their own way of differentiating a bit to certain directions. Some of them have different colors, some of them have different flavor compounds, some of them have a bit different profile of their chemical composition. So why did some of the cell cultures from strawberry turn out green and some red and then different flavor? We don't really know. I mean, plants are genetically way more complicated than we as animals are. And we don't fully understand how they work yet. That's work ongoing. let's figure out first the microbes and their, in our lives, then the animal cells, and then we get to plant cells and their, universe.
Yeah. I like this one too. Do different plant cell lines require different media? Some animal cell lines require different media. So what about plants?
Sure. Some cell lines do require a bit different kind of media, different kind of pH, for example, different kind of balance in the nutrients. Some of them require some light. Some of them don't. We have a really nice birch tree cell line that grows this purple-red color if it gets light. But as soon as you put it in dark, it's completely white. Then you give it more light and it's purple again. I mean, that's how plants do. So they are somewhat picky with the environment where they grow in, and the media needs to be slightly different to some cell cultures.
All right.
Coconut water is for very special occasions, like when we do these protoplasts, taking the cell walls away from the plant cells, then they are a bit more. You have to treat them very gently and give them a bit of coconut water, and then they're happy.
Well, very nice. Well, on that note, I'm going to thank you for your talk. Thank you so much for answering all these questions for us.
Thank you.